Welcome, Bio 340 students. My name is Robert, and here along with your other ATAs, and we'll be going over exam 3 review. So we'll also be posting this review session online right afterwards. We'll be going over some examples, and we'll allow you to ask some questions anytime you like. And we'll also have an answer session at the end. So we'll go ahead and begin. I'm covering DNA structure. And just as a review, some of the main experiments that led up to determine that DNA was the hereditary material were, one was the Frederick Griffith experiment. And they showed that non-virulent bacteria was able to be transformed by heat inactivated virulent bacteria. They used that with the mouse experiments, and they used a smooth coated bacteria and a rough coated bacteria. And then there was the Avery McLeod-McCarty experiment. And this one, the heat killed virulent bacteria, and they used that lysate, and they treated it with various enzymes. And the one treated with DNAs was the one where the non-virulent bacteria was observed. And the Hershey-Chase experiment, in which radioactive labeling of DNA and the protein coded phage were used to determine that the DNA was the hereditary material that infected the host cell. So these are the main experiments that led up to determine that DNA was hereditary material. So we'll go on. And just to recap, for DNA structure, there's the 4 nucleotides of DNA. There's adenine, guanine, thymine, and cytosine. And RNA has uracil. And so the main makeup of a nucleotide is there's the nitrogenous base, which can be any of those that were just mentioned, and that's attached to the sugar and a phosphate group. And that's the basic structure of a nucleotide. The one that's going to be interchangeable is that nitrogenous base. We'll go on. And then, so we have the pyrimidines and the purines, and one will pair with the other. The pyrimidines are the cytosine, thymine, and uracil, and those have a single ring structure. The purines are adenine and guanine, and those have a double ring structure. And we'll move next to the next slide. So base pairing is made by hydrogen bonds. Hydrogen bonds on a hydrogen or nitrogen will be the donors, and an acceptor will be on a nitrogen or oxygen-free electrons. One thing to mention is that thymine and adenine will form 2 bonds, whereas guanine and cytosine will form 3 bonds. Based on these bonds, some base pairing will be stronger than others. Guanine and cytosine will form stronger bonds than adenine and thymine because they have 3 bonds. And regions in your DNA where there's A and T rich regions will break apart easier than G and C rich regions. So here's a little application. When designing a PCR protocol for amplification of a region of DNA, which DNA sequence would require a higher melting point? So I'll let anybody say what they think based on these 2 strands of sequences of DNA. Which one would require higher melting point, or in other words, which one would be a little bit harder to break up? Great. So we'll move to the next slide, and I'll show that. So yes, the top sequence is going to have a lot more G and C bases whereas the bottom one will have fewer. So that top strand will require more energy to break apart those three hydrogen bonds between C and G. So another thing to mention about the DNA structure is that both strands of a DNA molecule are antiparallel. They run in opposite directions from 5' to 3', and the bases pair according to Chargaff's rule, with A binding with T and G and C. And here's another application. On a recent trip to Mars, you discover a series of artifacts, and you want to determine if any of them previously harbored any life, assuming that all life only has DNA. So according to Chargaff's rule, which is a potential candidate for further analysis? So sample 1 contains mostly guanine and adenine, about 40% each. Sample 2 has an equal amount of adenine to uracil, in addition to equal amounts of cytosine and guanine. And sample 3 has only guanine, thymine, and cytosine, no adenine. Sample 4 contained guanine and cytosine in equal amounts, in addition to adenine and thymine in equal amounts. So which one would be a potential rate? So sample 4, it wouldn't be sample 1 because adenine and guanine are in equal amounts, and according to Chargaff's rule, guanine will pair in equal amounts to cytosine, and there wouldn't be, so those numbers would not make sense. Sample 2 has uracil, so we know that DNA does not have uracil, so we can't look at sample 2. Sample 3 does not have adenine at all, so that's not a possible candidate. And sample 4 is the best candidate, has equal amounts of adenine to thymine and equal to cytosine and guanine. And that's it for me.
All right, guys, we're going to go over DNA replication, just some key things to remember. So in regards to the origin and directionality of DNA replication, most DNA replication is bidirectionary, meaning that it's going to be going in both directions from the origin of replication. For bacteria chromosomes, you're going to have only a single origin of replication. And then in this image right below is the eukaryotic chromosomes, where they have multiple origin of replications. And here's going to be 2 slides of important molecules for you guys to remember in regards to DNA replication. So DNA-dependent DNA polymerase 3. And this is the main replicating enzyme. And this has 3' to 5' exonuclease activity for proofreading. And just something to remember is the name, the polymerase 3, is just try to remember that it goes from the 3' to the 5'. That's just like kind of how I memorized it. And it is template dependent and primer dependent. And then primase, it lays down a small piece of RNA, which acts as a primer for DNA polymerase. Helicase, which is unwinds DNA at the fork of DNA synthesis. And Okazaki, these are fragments. And the way I memorized that was just kind of like the weird way of how I say that, which was just Okazaki, just broken up. And these will be on the lagging strand, where the DNA replication is on the opposite direction of the helicase. And then we have the ligase, which will join Okazaki fragments together. DNA-dependent DNA polymerase 1, which binds to a single-stranded gap, and it has 2 activities. First activity is it has 5' to 3' exonuclease activity for removing RNA primers. And then it'll add DNA nucleotides from the 5' to the 3'. And it is also template dependent and primer dependent as well. And then we have telomere, which is at the end of the chromosome. And then telomerase, which is, it repairs the ends of the chromosome. And it has an RNA dependent DNA polymerase activity. And then here's going to be the steps of DNA replication. So step one, the helicase breaks hydrogen bonds and unzips the DNA with topoisomerase, which is ahead of the helicase, preventing overwinding. Number two, the single-stranded binding protein, SSB, prevents the broken bonds from recombining. And then DNAG synthesizes the RNA primers, which is in red right there. DNA polymerase 3 synthesizes daughter strands once it is primed. And that's a key thing to remember. DNA polymerase 3 elongates the leading strand continuously, which is going in the same direction as the helicase. And then the lagging strand, it'll be discontinuous, and that's where you'll see the Okazaki fragments. Number 6, DNA polymerase 1 removes the RNA and replaces it with nucleotides. And number 7, DNA ligase replaces polymerase 1 and joins those Okazaki fragments by catalyzing the formation of phosphodiester bonds and sealing the gaps. And then just kind of like a quick review, if you guys can go ahead and identify A, B, and color coordinate it for you guys. Group chat for what A, go down that. Yep. Yeah, Savannah's got it, perfect. Next slide. Yep, helicase, leading strand, and Okazaki fragments. Perfect.
Hi everyone, I'm going to go over just a few slides on mutations, proofreading and repair. The first 3 slides that I have are very interactive, so I'm going to ask questions, so you can either unmute yourself or you can type in the chat box. So I have the 3 different types of mutations on the top: silent, missense, and nonsense. So looking at the answers on the bottom, which one do you think that silent would go with? Think of it as from left to right, look at it as A, B, and C, and tell me which one do you think that silent would match to? Perfect. Can you show the answer? 1 click. Perfect. B, a base pair change that does not alter the resulting amino acid due to the redundancy of genetic code. How about missense? Perfect. C, can you show the answer? A base pair change that results in an amino acid change in the protein. And then last but not least, go ahead and show the answer for nonsense. So that's a base pair change that creates a stop codon in place of a codon specifically an amino acid. Go ahead and go to the next slide. All right, so transitional versus transversional mutation. So before you go into detail on this, can anybody tell me what adenine and guanine are? What specific type of nucleotides they are? Perfect, go ahead and show the answer. So they're purines. When you look at them, you can identify them easily because they are the 2-ring nucleotides. So how about cytosine and thymine? Perfect. Go and show the answer. Pyrimidine. So they have the 1 ring. So I remember this kind of as an opposite. The bigger ones with the 2 rings have the smaller name, and the smaller ones with the 1 ring have the 1 name. So when you're looking at this diagram that I put in there, you can see the different colors of the lines connecting them. Can anybody tell me what the red lines stand for? Perfect. So Rebecca got that perfectly. Can you give me 1 click? So transitions are interchanges of the 2-ring purines or the one-ring pyrimidines. I'm sorry, I can't talk. And then give me one more click. And the transversions are interchanges of a 2-ring purine for a 1-ring pyrimidine or vice versa. Go ahead and go to the next slide for me. All right, so other types of mutations on here, I have frame shift and regulatory. Can anybody tell me the 2 different type of frame shift mutations? Oh, Savannah, so you put on there, so transition is a transition from one purine to another. That's a perfect way of explaining that. And then Savannah, yes, deletion and insertion. Can you go ahead and give me a click? So the frame shift is an insertion or deletion of one or more base pairs that leads to the addition or subtraction of mRNA nucleotides. This alters the reading frame of the message and provides the wrong amino acid sequence. Can anybody tell me the 3 different types of regulatory mutations? Okay, go ahead and give me a click. Oh, perfect. Actually, we just kind of slow. So they can alter the amount of protein product produced by a gene. This can affect regions such as promoters, introns in the regions coding for the 5' to 3' segments. So these are promoter, splicing, and cryptic splicing, or splice slides. So I didn't include all of the information, all of the pictures for all those, because it would have been a lot of slides. But if you go back to Dr. Holacek's lecture on the mutations, proofreading, and there's slides, I'm trying to remember which numbers they are, but there are 3 specific slides that go into great detail about these specific types of mutations, and it actually has the pictures so that you can kind of put what the name of they are compared to what they do. Go ahead and go to the next slide. So proofreading and repair. I actually found this little diagram here on Khan Academy. There's a really, really good overview of proofreading and repair. So this just is kind of a basic explanation of it. So when polymerase is going down the line and it adds in an incorrect nucleotide on the new strand of DNA, it detects that, oops, it put in the wrong nucleotide. So it's going to use the 3' to 5' exonuclease activity to remove the incorrect nucleotide and place in a new one. Go ahead and click the slide. So I think of them as a DNA spell check. Even though it kind of did the mistake itself, it still goes back and tries to fix it when something is wrong to kind of repair it and prevent these mutations from happening. Go ahead and click it. But sometimes it makes mistakes and it gives longer segments of this mistake. So go ahead and give me another click. Well, why is it necessary if there is a proofreader? One more click. Sometimes the proofreader makes mistakes and larger components of DNA needs to be fixed after proofreading has been completed. So when a mismatch is detected in newly synthesized DNA, the new DNA strand is cut and the mispaired nucleotide and its neighbors are removed. The missing patch is replaced with the correct nucleotide by DNA polymerase, and I always say that wrong. The DNA ligase seals the gap and the DNA in the DNA backbone, so it kind of repairs itself. So there's a little more information that was covered in the slides, and I would suggest going into it because it's a lot of vocabulary on learning what kind of things promote these different types of mutations, like whether it's a nucleotide-based analog, whether it's an oxidizing agent, and then there's also information in there about radiation-induced DNA. So I would suggest that you just go in and kind of look over that, because most of this area, besides knowing how the repair is completed, is going to be vocabulary for you. And that's it. Thank you.
Okay, so I'm going to be going over transcription. I think with these modules, we kind of got more in depth over, how transcription really works. So it varies between prokaryotic and eukaryotic organisms, as we learned, but they both have the basic processes of initiation, elongation, and termination. And don't forget to know the difference between your template and coding strand. Up here, we have the arrows drawn. So your template strand will run 3' to 5', and your coding strand will run 5' to 3'. And notice that the sequence of the coding strand is identical to the mRNA, but with, the U's in replace of the T's. And so transcription is going to be controlled by specialized regions of DNA called promoters. So your promoter regions will be different between prokaryotic and eukaryotic organisms. And transcription will only begin when RNA polymerase binds to the promoter and then begins to unwind the DNA double helix. The template strand will then come in and build a complementary RNA strand. So getting into specifically prokaryotes, their initiation begins when RNA polymerase recognizes and binds to the gene's promoter. And the promoter region is characterized by this downstream, maybe this is upstream, 35 region consensus sequence. So your 5' to 3' TTGACA. And then your PribNow box at around the 10 base pair region, which is TATAAT. It's important to know that this PribNow box is going to be AT rich. Then we'll go into elongation, where RNA polymerase will move down the gene, unwind the double helix, and build the mRNA and rewind the double helix behind it. And then termination, we have RNA polymerase coming in, recognizing a specific nucleotide sequence, causing it to just stop transcription. And the terminator region of the mRNA will fold into a hairpin structure, like I have in this picture right above here. You're going to have a string of uracils after the hairpin. And unlike eukaryotic transcription, there's going to be no addition of a 5' cap, no addition of a 3' poly A tail, and no splicing of pre-mRNA. And in termination, there are inverted repeat sequences in the transcript that fold into a complementary stem separated by a single-stranded loop. And then hydrogen bonds between the A and the U base pairs break, which releases the transcript and then terminates transcription. So going into eukaryotes now, there's actually 3 polymerases, each made of about 10 plus subunits from mRNA synthesis. So you have RNA polymerase 1, which transcribes ribosomal RNA genes. RNA polymerase 2 transcribes all protein coding genes into messenger RNA. This polymerase 2 will actually make the pre-mRNA, and that undergoes co-transcriptional modification before leaving the nucleus, which we'll get more into when we talk about termination for transcription in eukaryotes, but this involves the addition of a 5' cap, the addition of a poly A tail, and splicing to remove introns and join exons. And last but not least, RNA polymerase 3 transcribes transfer RNAs and other small functional RNAs. So initiation will begin, the TATA binding protein, which is a general transcription factor, will bind to the TATA box. And so once that binding protein is bound, other transcription factors and RNA polymerase 2 will join in and form the pre-initiation complex. So important promoter regions to know here, you have your GC-rich box, your CAAT box, and your TATA box. So I'm going to check the chat here. I see some question. Okay. So elongation is RNA polymerase 2 will separate from the general transcription factors, move down the gene, and make RNA. And the transcription factors become available to initiate transcription by another RNA polymerase 2 complex. And finally, we have termination, which is where the eukaryotic pre-mRNA undergoes the extensive processing that we talked about here involving RNA polymerase 2. So You have your 5' cap, which protects RNA, and it's needed to initiate translation of mRNA to protein. Transcription will officially end when the poly A tail is identified. The termination signal sequence is AAU, AAA, and intervening sequences not coding for protein, which are introns, are going to be removed or spliced from protein synthesis. And going back to that poly A tail, an endonuclease recognizes the poly A tail and will cut the RNA about 20 base pairs downstream.
All right, so I'm going to be talking about translation. So we're going to bring it back to the central dogma of molecular biology. So you can see that transcription is from the gene of a DNA to the transcript of an RNA, and the process of translation is from the transcript of RNA to a polypeptide, which is composed of amino acids, which compose proteins. So what is translation? It is the process in which ribosomes in the cytoplasm or endoplasmic reticulum synthesize the proteins after the process of transcription of DNA to RNA in the cell's nucleus. So this kind of whole process is called gene expression, and gene expression is the steps through which a gene influences certain phenotypes. So as you can see in the diagram on the very bottom, the DNA has a non-coding strand, which is a template strand, and then it has a coding strand, which is known as a non-template strand. And then transcription consists of the RNA, and then translation consists of the protein. As you can see, they're on the right side of the diagram. Next slide, please. So what are proteins? Proteins are the polymers made up of building blocks of amino acids, and each protein consists of about 20 amino acids. So based on their side groups, their structure and the chemical properties differ between all of them. So one protein is composed of an amino group, a carboxyl group, and then a side group. Next slide, please. So as you can see here, these are some examples of non-polar side chains, electrically charged side chains. So these are just important ones to remember for your exam. They're written on this diagram. I would probably just go through this and just look through all of that. Next slide, please. So what are polypeptides? Polypeptides are chains of amino acids that are linked together by peptide bonds. It's a linear organic polymer consisting of a large number of amino acid residues, and they're bonded together in kind of a chain-like formation. So it forms a part of or the whole part of a protein molecule. So in the diagram on the bottom, you can see the amino end and the carboxyl end, and it also contains amino acid 1, amino acid 2, amino acid 3, and they both have peptide bonds between each of them. Next slide, please. So what makes up a protein? So protein structures consist of 4 parts: the primary structure, which is the chain of amino acids and the alpha helix, the secondary structure, which is the pleated sheet, the tertiary structure, and then the quaternary structure, which is the hemoglobin globular protein. So the genetic code is kind of produced from a non-overlapping triplet code. So 3-letter codes represent how many amino acids are in the sequence. So like in the example on the very bottom, I gave an example of ATG, ATC, TGC, TGA, and continued. And to kind of solve this and see how many amino acids are in there, you can see that there's 4 individual ones, and each one is composed of 3. So for this kind of thing, you'd have to do 4 times 4 times 4, and that would give you 64. So the genetic code and the gene structure. The genetic code is seen to be degenerate. So degeneracy means that 2/3 of mutations will be silent mutations, and it even occurs in like the coding regions of a genome. And so there are 64 more codons than amino acids in this. So as you can see on the very right side, there is a gene structure that shows DNA and mRNA. So in the DNA, there is a template strand going from the 3' to 5', and then a coding strand going from 5' to 3'. And then the mRNA is consisted of a UTR, which is an untranslated region, and it also goes from the 5' to the 3'. So the coding sequences, they have start and stop codons. So it consists of an open reading frame, also known as an ORF, and it gets translated into a polypeptide. So it first starts off with the start codon. This encodes the polypeptide's first amino acid. And as you guys already know, the first normal start codon is AUG and encodes the amino acid methionine. And the open reading frame usually ends with the stop codon. So the 3 normal ones that we usually see are UAA, UAG, and then UGA. But these 3 stop codons do not encode amino acids, but rather tells the protein synthesis machinery to stop translating it. So in between each of the stop and the start codon series, in between them, there's a series of triplet codons, and each one of them specifies a specific amino acid. As you can see here, it goes from the 5' to the 3' side. And then this is the chart that's going to help you just kind of navigate and look through all of that. So next we're talking about translation machinery and tRNA. So mRNA are also known as codons. So then they also have ribosomes or ribosomal RNA, which is composed of ribosome RNA and ribosomal proteins. So the tRNA and the tRNA charging enzymes, it's also known as amino acid tRNA synthesis. And the transfer tRNA are also known as adapter molecules, and they match codons with the correct amino acids. So a tRNA is usually about 74-95 base pairs in length, and it folds into kind of a cloverleaf shape. So each tRNA only binds to one of the amino acids, and each anticodon is complementary to one codon for its certain amino acid. And we're going to be talking about the degeneration of tRNA and ribosomes. So most amino acids can be brought to the ribosome by alternative tRNA. This means that several tRNA can carry the same amino acid, and this is known as the wobble effect. So the same tRNA interacts with different kinds of codons. And then about ribosomes, ribosomes are extremely abundant within living cells. So as cells expand, more energy and protein synthesis and any other activity. So the protein synthesis machinery makes up about 1/3 of the dry mass of the cell. And most of this dry mass of the cell consists of ribosomes. So most of the RNA within the cell is also known as the rRNA. So with ribosome binding sites in tRNA, it consists of 3 parts. So the E means exit. So they're only present in large subunits, and it provides an avenue for exit of the tRNA after its amino acid has been added to the chain. So the P side stands for peptidyl, and it holds the tRNA to which the polypeptide is attached. And then there is an A part, which is the amino acid, which binds a new tRNA molecule containing the amino acid to be added into the growing polypeptide chain. So a specific type of sequence that we're going to be looking over is the Shine-Dalgarno sequence. And this sequence is complementary to an rRNA sequence in the 30S subunit. So in certain prokaryotes, this special sequence is in the 5' UTR, and it helps the ribosome find the start codon in mRNA. So the complementary bases pair up, the start codon is placed in the correct position. So as you can see here, it's the Shine-Dalgarno sequence is the AGGA-GGU, and there is a start codon methionine, which is the AUG, and it contains a 5' UTR in the coding sequence. So now we're talking about the initiation in prokaryotes and eukaryotes. So in a prokaryotic mRNA, it goes from the 5' to 3', and it contains that specific Shine-Dalgarno sequence. And then in eukaryotic mRNA, it contains a 5' cap, and this is where ribosome scanning occurs from AUG to another AUG to another 5' cap. So this kind of diagram on the right is just very important when it talks about the 30S subunits and about the initiation factors and how GTP binds to the 30S ribosomal subunits. So the next rule is Kozak's rule. So this is where there's certain steps on how the ribosomes bind and form the cap structure. So in the first part, the 40S binds the 5' end of the cap structure. And then in step 2, the 40S migrates on the 5' UTR until it gets to the optimum start codon AUG. And then the 60S will join the 40S subunit. And then the ribosomal complex will start the process of translation. So back to the initiation in eukaryotes, the initiator tRNA is charged with a modified methionine, which is also called FMET, and it should be brought to the P site. So when the mRNA is positioned so the start codon is at the P site, the ribosome has about 2 key regions that help to elongate the polypeptide. So it contains a decoding center, which is in the small ribosomal subunit, and it ensures that only a small complementary tRNA part is allowed into the A site. And then it also contains a peptidyl transferase center, and it's in the large ribosomal subunit, and it catalyzes the formation of peptide bonds. So as you can see in this diagram here, so it shows the third charge tRNA has entered the A site, so it enters through here, and is facilitated by the EFT, which is the second elongation step begins. And then this is where the tripeptide is formed, the second elongation step is completed, and the uncharged tRNA moves to the E site. And then the very last step is when the polypeptide chain becomes synthesized and it enters the existing ribosomes. So the peptide bond formation it contains the 3 termination codons UAG, UGA, and UAA, and it contains certain GTP dependent release factors, and it binds to the stop codon, and it cleaves the polypeptide chain from the tRNA and releases it from the translation complex. And this is where the dissociation of ribosomes and eukaryotes occur. And that's the end of my part. Thank you.
All right. Let's talk about the transcription control of gene expression. So we have prokaryotic and eukaryotic organisms. So the gene expression will be different in each of them. The prokaryotic transcription and translation occur simultaneously in the cytoplasm, and the regulation occurs at the transcriptional level. Now, eukaryotic gene expression is regulated during the transcription and RNA processing, which takes place in the nucleus, and during the protein translation, which takes place in the cytoplasm. You see a cell's ability to control which genes are active under any given environmental conditions is very critical for its function. So such controls allows cells to choose whether to utilize nutrients available in the environment, or if a needed nutrient isn't present in the environment, for example, it is able to manufacture it via an enzymatic pathway if it's able to do so. So in order to control the products it manufactures at any given point in the development or its life cycle, a cell basically must be able to react to the feedback mechanism that will allow it to do two things. One is to repress the manufacture of a constantly made product, or to induce, basically to manufacture the product that is not constantly made. So you would wonder how, right? So therefore, basically the cells need mechanisms that allows for two things. One of them is to allow them to recognize specific environmental conditions where will require them to either activate or repress the transcription of the genes relevant under those conditions. And the second is to allow them to turn the genes basically on or off as needed. Next slide. I just wanted to go over quickly over some terminology. So we have, as you'll see at the top picture, we have operon. And you will see it's basically a sequence of the genes adjacent to one another on the DNA. And under the collective, the operon is basically under the collective control of a promoter and an operator, and co-transcribed as a single mRNA. So then we have a promoter, you see that in orange at the top. And this is basically a region to which RNA polymerase attaches at the initiation of the transcription. Then right after that, we have the operator. It's a short DNA sequence, usually just upstream of the promoter. And then if you look to your left, we have positive and negative control. So positive control, or sometimes it's said when the transcription is under positive control, it basically means a protein known as an activator. It is right at the top in the purple. It binds to the DNA in order to for the transcription to take place. And then we have a negative control, and that's slightly at the bottom. You have the purple repressor. You see in the picture, and that is basically a protein that when it binds to the DNA, it will block transcription. Can we go to the next slide? All right, so gene expression systems may operate under either positive or negative control, as mentioned earlier. Each type is often linked to inducible and repressible systems described above. So a classic example of that would be, for example, found in E. coli. We have the lac operon, which will be the inducible system, and then oh my God, I forgot the word, but it's a TRP operon. And repressible, it's in the repressible system. So you would wonder, what is the difference between the positive and negative regulation of the gene expression, right? So Positive gene regulation allows the production of a gene that is needed for the use at a particular time or situation in the cell. However, the negative gene regulation prevents basically the overproduction of that gene at a certain time, right? So if you look at the picture right on the top right, you'll see that the positive control, the genes are expressed only when an active regulator protein, basically called activator, you'll see that in the purple, when it's present, therefore the operon will be turned off when the positive regulatory protein is absent or inactivated. And then vice versa, when in a negative control, we need to have the genes in that operon will be expressed unless they're switched off by this protein, repressor protein. You'll see that in purple as well. Therefore, the operon will be turned off, and the gene will be expressed when the repressor is activated. Can we go to the next slide? So here I have a couple questions, and the question is, which of the following mechanisms are used to coordinate the expression of multiple related genes in eukaryotic cells? And if you guys have any guesses, educated guesses? No? Okay, that's okay. Can we get a next slide? So we got AOD. So in this case, the answer is B. Genes share a single common enhancer, which allows appropriate activators to turn on their transcription at the same time. You know, the enhancer basically is like the short region of the DNA that can increase transcription of a gene. They can also be, they are located upstream of a gene, which coding within the coding region of a gene, or sometimes downstream of a gene, and is basically the enhancers. They promote transcription. Each enhancer is made up of that short DNA sequences. They often called, they are called, you'll see on the picture on the top here, distal control elements. And the activators bound to the distal control element interact and with the mediator proteins and transcription factors, and therefore they kind of like stimulate that transcription. All right, can we go to the next slide? And another question I have is, control of gene expression in eukaryotic cells occurs at which level? What will be your guess? Yes, D is correct. D is correct, yes. And it's interesting that in eukaryotic cells, the regulation may occur when the DNA is uncoiled or loosened from the nucleosomes, and you'll see that on the picture to bind the transcription factor. That's basically epigenetic level. And then transcriptional level will be when the RNA is transcribed. And then the post-transcriptional level is when RNA is processed and exported to the cytoplasm after it's basically transcription. And then our post-translational level would be when the RNA basically is translated into the protein. And then after that, protein has been made, basically produced. However, just to know that in prokaryotic cells, the control of gene expression is mostly at transcription level. So that's where it stays. This is the end of my slides.
Okay, so I'm gonna go over the lac operon. So this is just an overview of the lac operon, which is used for the metabolism of lactose. So you have areas like your repressor, which is your LAC-I gene. This is an example of a trans-regulatory element, meaning that it can regulate a gene that can be on a different chromosome. And you have areas like your promoter and your operator, which are cis-regulatory elements, meaning that they regulate genes that are on the same chromosome they are on. Next slide, please. Okay. So lac operon can be controlled through both positive and negative control. Negative control essentially means preventing gene transcription. So in the lac operon, lac-I produces a repressor protein. This repressor protein can bind to an operator, and this prevents transcription. So the repressor protein can also bind to allolactose. This is called the inducer, and this causes the repressor protein to no longer be able to bind to the operator. So this allows for transcription. So essentially, the presence of lactose is going to be turning the gene on. And lac operons also have positive control, which increases the levels of gene transcription. So essentially, the body prefers glucose as a fuel source, but sometimes when there's not enough glucose, we need to be able to use the lactose. So when glucose levels are low, we have our cyclic AMP that's going to build up into the cell. And the cyclic AMP is going to bind to our cap, creating a cyclic AMP cap complex. This binds to our DNA and allows transcription. Next slide, please. Okay, so this is a super useful chart that I recommend you look at, you study before your exam. It goes over different kinds of protein-producing genes, their products, and what these products do. So LAC-O and LAC-P are pretty straightforward. LAC-I is what was producing your repressor protein that we were just talking about. LAC-Z is producing your beta-galactosidase, which cleaves your lactose. LAC-Y produces your permease. This helps your lactose be able to get across your cell membrane. LAC-A produces your transacase, which essentially protects the cell from the byproduct of this lactose metabolism. Next slide. Okay. So I think this should answer your question, Yash, but let me know if it does. It's really important to know when the lac operon is functioning and how it is controlled. So when glucose is present and there's no lactose, your repressor protein binds the operator, there's transcription. When glucose and lactose are present together, your allolactose, so your inducer, is going to bind to your repressor. Your repressor cannot bind to the operator, so that's going to create a very low level of basal transcription. When you only have lactose, your allolactose still binds to the repressor cannot bind to the operator, but then you get your CAP, cyclic AMP complex that we were just talking about, and this binder activates your transcription. Okay. 1 second to comments. Okay, see, transcription is just kind of very, very low level of transcription as compared to cyclic P and CAP. Their job is to really increase the transcription that's happening. Okay, next slide, please. Okay, so this is an example problem. So what would you expect to see with a cell that has this phenotype? And you can just throw your answers into the chat. Okay. So this can be kind of confusing, so we're going to break it in a little. But LAC-I is responsible for being a repressor protein, right? So if you mutate your repressor protein, it's not going to bind to your operator correctly. Then you're going to have constitutively expressing or constitutively transcribe something. And this means whether or not lactose is present, you will be having transcription. So LAC-Z responsible for LAC-Z responsible for creating beta-galactosidase. So mutate LAC-Z, you have no creation of beta-galactosidase. So we know it has to be constitutively expressed. There's no beta-galactosidase. Yeah, so D is the correct answer. Good job. Okay. This is another question. So if you're given a cell and you're told that this you're experimenting on type XGAL experiment, and the color is blue even without. So was the gene you were given for the cell correct or incorrect? And if it was incorrect, what would the correct genotype be? Okay, yeah. So we already talked about LAC-C from our previous question. LAC-C is going to be creating your beta-galactosidase. What does your blue color indicate? Okay, so blue color indicates that your beta-galactosidase is present. So for producing product, we know that it has to be functional, so it cannot be mutated. This gives us an indication C could be our correct answer. And we also know for O, mutation there will result in the repressor not being able to bind to the operator. So that will give you constitutive transcription. So even without the presence of lactose, beta-galactosidase, and it's still going to be turning the color blue. Okay. That was it.
Okay, so can everyone hear me okay? Okay, cool. So I'm going to be going over PCR, also known as polymerase chain reaction, which is a really common method used in biomedical labs. And I think Dr. Holjek mentioned that it's also used in forensics to identify suspects and things like that. So it's basically just a method that's used to amplify DNA segments exponentially. So remember that. And it uses the principles of DNA replication by using these different reagents. So a double-stranded DNA template with a target DNA sequence, DNTP, so those are just going to be your different, the full name for it is DDO nucleotides, which are just your adenine, guanine, cytosine, and thymine. And then you're going to use a heat-stable DNA polymerase. A lot of the times people will use the Taq polymerase, and this Taq polymerase just comes from a heat-resistant bacteria. So it's able to undergo all the high temperatures that you'll use in a PCR protocol. You'll also need forward and reverse primers that are gonna anneal to your target region of DNA. And you also need a buffer. Next slide, please. All right, so on the right-hand side on the picture that I included are the different components that I already mentioned. So once you're done preparing your PCR reactions, you're gonna go ahead and put them in this machine called the thermal cycler. And then it's gonna go through this PCR cycle that I'm on the left-hand side for a total of 35 cycles. So first it's gonna be a hot start, and this just denatures the DNA. And then after that, you're gonna do the actual denaturation. So the hot start is just kind of like it's kind of like you're heat shocking the DNA. And I know you went through this in class, so this should be review. And then the third step is just you're gonna be annealing the primers that we mentioned before to your template DNA. And using the principles of DNA replication during the extension step, you're just gonna extend the primers that already annealed to your original DNA sequence. Usually these are gonna be complementary to a specific target DNA sequence that you're gonna wanna amplify for whatever experiments you're running in the lab. So, so you're gonna design certain primers that are gonna anneal to the specific DNA sequence that you want and amplify that specific DNA sequence. So the forward primer that you're using is always gonna be identical to your coding strand, and the reverse primer is complementary to your coding strand. And right below those two bullet points, I kind of did a visual representation of what your different primers would look like compared to the template DNA. And then to calculate what temperature you're going to set the PCR protocol at, you can use the Wallace rule, which I put on the left-hand side, or sorry, the right-hand side of the slide underneath the picture. So that's just an equation you would use. Next slide, please. Okay, so after you're done running your PCR, generally you can use your PCR products to run different experiments, and we learned about one of them, which was gel electrophoresis. So after you've amplified your target sequence, you can separate the different DNA fragments that you got by size using gel electrophoresis. So when you're looking at a gel, you're going to set it up with the negative electrode at the top of the gel. Or in this case, if you look at the picture, it's on the left-hand side, and then your positive electrode is on the right-hand side. And since it's based on size and it's a porous gel matrix, the smaller DNA fragments are going to go through those pores quicker because they're smaller, right? And since DNA is negatively charged, they're going to go towards the positive electrode. So that's how you get the separation of different DNA fragments. And then on the next slide, we're going to talk about another assay that we can use in the lab. It's called deoxynucleotide sequencing. So yeah, you can go ahead and go to the next slide, thanks. So it's just you have your different, let's say you have different PCR products and you want to know what the sequence of those are. So you can run a gel, and based on the separation of bands on the gel, you can kind of tell what the DNA sequence that you have is. So for this question, this was on your review, so some of you might know how to do it already. So it's asking, what is the sequence of DNA strand being synthesized during sequencing? So does anyone know, or does anyone know how to start off answering that question? Or I can go ahead and do it for you guys. You can type in the chat or just unmute yourself. Okay, yeah, so I think Sarah got it. Kind of, I put the arrow there for you. So if you ever see a question like this, all you're gonna do is you're gonna look at the different columns. And so you can see that the first one is gonna be the adenine, and then it's thymine, cytosine, guanine. So you're just gonna read the gel from the bottom up. So I put the arrow there for you. So it's pretty straightforward. You can see that the lowest band on the gel is going to be a thymine. And then if you follow the arrow, you have adenine, guanine, and so on. And then you can just, so you start from the bottom because that's where the smallest fragments migrate. And I'm not sure if that means, yeah, so that's going to mean generally, and go ahead and click the slide. I added a different diagram. Yeah, so basically that's going to mean that that's your 5' end. And remember, you always read from 5' to 3'. So that's why it's from the bottom. And then if you click again, it's going to, I typed in the actual sequence that you should get from this gel. So go ahead and click one more time. All right, and so that's it for that question. Next slide, please. All right, so this is a different type of, so this is called recombinant DNA technology. This is completely different, but it's also really common in different biomed labs. So this just means that we're artificially combining DNA from 2 different organisms. So you'll usually have the host species and then DNA from a foreign species, so foreign DNA. And then once you've combined both, then you get a transgenic organism. This is the host of the new DNA from the different species. And we have 3 different types of horizontal gene transfer that we use in the lab. So the first one is going to be conjugation, and this is the transfer of replicated DNA to a recipient. So you can see that in the picture. There's a nice little diagram. Usually, this is using bacteria. As you can see, it has plasmid DNA, which is bacterial DNA. And so you use the DNA that this bacteria synthesizes, and you pass it on by conjugation to the recipient organism. You also have transformation. Transformation is really common in the lab too. You'll usually use the plasmid DNA, so circular DNA, and then vector DNA that comes from the environment. So as you can see in the picture, you have a different bacteria that's been lysed and it's dead, and it transfers its genes to a different bacteria that's competent. And competent just means that we can culture this bacteria and have it grow colonies with the new recombinant DNA. The last type of horizontal genes transfer is transduction, and this is transfer of DNA from one bacteria to a different one, but using viruses. Next slide, please. Okay, so I'm gonna go over molecular cloning. This uses the principles of transformation. And some different things that you would need for this are a source of DNA from usually a vector, a linear vector that used to be a plasmid, but usually people will do a restriction digest using restriction enzymes. And that's another thing that you're going to need for molecular cloning. This just cuts the DNA and makes sticky ends. So the sticky ends are going to be different segments of DNA that contain an overhang and can be easily ligated with the enzyme ligase. So that's the next component that you're going to need for molecular cloning. This will just join the foreign DNA and the vector to make the recombinant DNA. And then you're going to need a host cell. Usually you'll use some type of bacteria. So as you can see in the picture, where it says transformation, usually after you've ligated the foreign DNA to the vector that you have, you'll usually plate it with a bacterial colony and then let this colony grow. So then you have a lot of that new recombinant DNA that you inserted into the bacterial colonies. Next slide, please. And that's it for my segment.
Okay, there should be a slide before this one. Okay, cool, thank you. So a lot of personal genomics relies on genetic testing, and genetic testing is a medical test or research tool that allows people to identify mutations, chromosomal abnormalities, insertions, deletions, substitutions. So essentially, a lot of what you guys have been talking about in class in terms of mutations, you can use genetic testing to identify. So when we look for these mutations, we actually want to look for mutations that could be disease causing. So there are many different kinds of genetic tests that we use. So you can use chromosomal genetic testing to look for larger changes to a chromosome. An example of this would be a karyotype, which you can see in the picture to the right. And you can see in this karyotype, you actually have an extra sex chromosome. So that's an example of kind of a larger change that you can see using chromosomal genetic tests. Molecular genetics looks at specific genes, or you can look at a panel of a larger variety of specific genes. And you can also analyze products of different genes by looking at their protein level to indicate if there's too much of a product being produced or too little. Next slide, please. Okay, so there's many different situations in which genetic testing may be useful. It's very, very important for diagnosis, and it's also very, very important for carrier screening, which you guys may remember about when we talked about methods of inheritance. Genetic testing can also be used in forensic settings. I know you guys talked about, I think it was the Golden State Killer in class. So they essentially were able to use his relative's DNA and trace it back to him. And you can also have things like newborn testing, preconception testing, pre-implementation testing, which you can use to decrease the risk of genetic abnormalities in future offspring. And you also have commercial testing, but this can be problematic if results are not easy to interpret, they're not easy to comprehend, or they're results that could be highly emotional for some people. Next slide, please. Okay, so you can use all of these different kinds of methods to study personal genomics or genetics even further. So SNPs are single nucleotide polymorphisms, and they're essentially a change to one nucleotide. So these can occur normally or benignly throughout a population, or they can be disease-causing, occurring in an individual or small group of individuals. And genetic testing yields the best results and has the most usefulness for making future health decisions when it can be applied with someone's individual medical and family history. So RSRSs are reconstructed sapiens reference sequences. So they analyze mitochondrial DNA, which is maternally inherited. Through using modern human and ancient hominid samples, we can actually trace people's ancestries through their maternal line. And genome-wide association studies have led to the discovery of genes linked to different disorders, but they also raise a lot of questions about risk factors. So essentially smaller or changes to genetics or changes to genes that have smaller effects but can build up to have a larger effect, and then the environment also plays a factor. Next slide, please. Okay, can you hit the button again? Okay, so what is the goal of pre-implantation testing? Okay. So pre-implantation testing essentially decreases the risk of genetic or chromosomal diseases or disorders. And it is often done when both parents are carriers for a heritable genetic disorder, and they want to ensure that their child will not get this disorder, or even that their child will not be a carrier of this disorder. Can you hit the button again? Okay, so if I wanted to determine my ancestry, which method would be most appropriate? And if you could hit the button again. Okay, just hit it twice more, sorry. Okay, so which one of these three methods would be most appropriate for determining my ancestry? Yeah, B is absolutely correct.
Okay, so now we're going to be talking about epigenetic inheritance, and if I'm correct, this is actually the last thing we're going to be talking about. So epigenetics is the study of potentially heritable changes in gene expression that does not involve the changes to the underlying DNA. So this can be influenced by many factors, including age, environment, lifestyle, and diseases. Epigenetic modifications can manifest as commonly as the manner in which cells terminally differentiate. So at least 3 systems are currently considered to initiate and sustain epigenetic change. To start, you need DNA methylation, and next you need histone modification, and then you also need non-coding RNA. We'll talk a little bit further about DNA methylation, but that condenses DNA. It has CpG dinucleotides, and it is symmetrical. Next slide. Okay, so I know that we already saw this slide earlier, but we can just, kind of walk through it maybe again. But basically, to start with epigenetic mechanisms are affected by many factors, and it's listed in the top left corner. So you have things like development, environmental chemicals, drugs and pharmaceuticals, aging, and diet. So this picture just shows basically a chromosome, which is made out of chromatin, which is made out of DNA. So DNA methylation shows that a methyl group can tag DNA and activate or repress the genes. And you can see that it's going to be like attached, but it does say that histones are proteins around which DNA can wind for compaction and gene regulation. And then if we move to the right a little bit more, you have the histone modification, which is shown in the right picture at the bottom right. And it says the binding of epigenetic factors to histone tails alters the extent of which DNA is wrapped around histones and the availability of genes in the DNA to be activated. And then in the top right corner, it says health endpoints. So this is like what this can cause, and that could cause cancer, autoimmune diseases, mental disorders, and diabetes. There's more, but those are very common. Next slide. Okay, so this is actually a picture. I, till this day, actually remember this picture. I took genetics my sophomore year, and I'm a senior now. And for some reason, this picture and Dr. Holchek telling me about this has always stuck with me. So the addition or the removal of methyl tags can be influenced by the environment. So that could include diet, exercise, stress, and chemicals. And basically what Dr. Holchek said is that, like, you know, you could be affecting, a grandmother could be affecting its grandchild based off of these methyl tags and what environment they surround themselves with. So you can see, like, the mother is the first generation, then you have the fetus, which is the second generation. And then within that fetus, you have reproductive cells, so the third generation. So that mother's, her environment is very important to her, not only her child, but also her grandchildren. Next slide. So we talked about the twin study case, which is a very common case. I'm sure many of you guys have even heard of it before Dr. Holchek went over it. But the twin studies uncover genetic and environmental contributions. So twins share the same genes, but their environments become more different as they age. So this unique aspect of twins makes them an excellent model for understanding how genes and environment contribute to certain traits. So if we look at the picture below, you have an inherited epigenetic predisposition. So then you have at the top, you have the normal twin and then the affected twin. So we'll look at the affected twin. You can see, like, that methylation, so epigenetic changes during development, which causes a reaching of the threshold. So that's the first episode of psychosis to remission to relapse. So there's plenty of like articles and stuff about these types of twin studies. It's really cool to check out. So I highly recommend it. Next slide. Okay, so now I just have a couple questions for you guys. You guys can just put in the chat. But the first one is, what is epigenetic inheritance? Okay, awesome. Yeah, it is A. And then the next one is true or false. Twins share the same genes and environment as they age. Awesome job. Yes. Actually, can we just go back to the previous slide? Okay, so I just really, really want to emphasize that first bullet point where it says twins share the same genes, but their environments become more different as they age. It's really, really important. Might show up on the exam. Very important just for future classes that you also take in biology. And with that being said, I think that's all we have for the review. If you guys have any questions, let us know in the chat or unmute yourself too.
Hey, Jean.
So this will be uploaded when, I think it'll be about at least an hour it usually takes to, like, be sent after we end the Zoom call. And then Dr. Holchek will send an announcement out to you guys, including the link.
Megan, Mark asked a question about whether twins share the same genes or not. Do you want to address that?
Yeah, sorry, I was just typing it, but they don't, correct me all any TAs here, they don't share the same genes. They say they share very similar genes, especially if you're identical.
Okay, Savannah wanted to go over design primers. Who is doing design primers?
I'm not sure if anyone really did. What is that like a part of? And I know someone asked why, where is it? Why does their cell still remain blue if lactose is not present? So I think going back to this question, who had this question?
That was my question. Yeah, so I can explain that. Who asked that? It was Yash. Okay, so essentially, if the color is still blue, even though the lactose is not present, we know number one, beta-galactosidase is being produced, right? Because we get the color blue from the beta-galactosidase. So beta-galactosidase's job is to cleave lactose and to create products that our body can then use. But even if we don't have lactose there, beta-galactosidase can still be present and it can still be causing the blue color if there is a mutation that essentially causes the beta-galactosidase. I think it was, I'm trying to remember the other, if it causes the beta-galactosidase and the other genes that are with the beta-galactosidase to be expressed constitutively. So if they're expressed constitutively, that means that whether or not lactose is there, they're always transcribing those genes. So we can have it blue if we're always transcribing our beta-galactosidase. Does that make sense, Yash? Okay, you're under the impression that blue is from hydrolysis of the lac. Okay, so essentially the color blue is part of your X-Gal experiment, and someone can correct me if I don't explain this well enough. But you're getting the color blue because something is being hydrolyzed in the beta-galactosidase, right? So without the beta-galactosidase there, your color would be clear. Okay, and Savannah wants to know if C minus means that, okay, so if you have lac C minus, that means there is a mutation in lac C, and that mutation can actually cause the lac C to be unable to transcribe your beta-galactosidase. Does that make sense, Savannah? Okay, good. Did anyone have any questions about the operon or the lac operon? I know that can be a really confusing part for some people. Sorry, Bridget, can you clarify your question? Okay, you were just piggybacking off Savannah's question. So essentially, if your LAC-C is mutated, this means that your LAC-C is not going to be functioning correctly. So if it's not functioning correctly, it's not going to be transcribing beta-galactosidase. Okay, so it is possible for it to be blue if, okay, so in this question, the color is blue because your O is being mutated to essentially make all of your products be constitutively expressed. You can also mutate your I in such a way that you can make all of the products constitutively expressed. What do you mean by a super repressor mutation, Ainsley?
The one where like it always binds, or oh wait, maybe I'm wrong. That's where the allolactose binds to the the thing that binds to the operon that initiates it, right? Oh wait, no, so that would be blue. Okay, I think I'm just remembering it wrong.
Okay, gotcha. Oh. So Yash wants to know if we had the mutation to O to make it constituent and Z positive with glucose present and lactose absent, would we still make beta-galactosidase or would we make none? Okay, so in that situation, because your LAC-O, your LAC operator, is constitutively expressing, you are still going to be making your beta-galactosidase. Because if you mutate your LAC-O to make it constitutively express, it does not matter whether the lactose is there. It does not matter whether the lactose is not there. It's going to express it no matter what. Okay. Did anyone have any other questions?
Okay, Robert. Oh, there's one more question.
Okay. Is the mutation to IS dominant? Give me 1 second. So IS essentially means that your allolactose can no longer bind to your repressor. So if you mutated your repressor to change your allolactose binding spot, so your repressor is then going to bind to your operator and there's going to be no transcription. So what did you mean by, is it dominant over what?
Like if we had a mutation IS in a vector and we introduced it to like a gene with I plus or I minus, like would the IS prevail, like its function, or?
I do not know 100% about that. I think, right, because your LAC-I is your only trans regulatory element. So it can essentially, if you introduce that on a vector, it could potentially move to whatever your bacterial DNA is, but I'm unsure if it would be dominant. Does anyone have any idea about that? Okay, Bridget is saying it's an activity and that I plus would allow for it to proceed. So I'm not sure which activity that was, but you can go back and look at that and stuff.
Okay, I would say, Robert, you can stop recording to get this video started on its processing.